Thanks, Megan. That was very kind. And I hope you all agree that Megan was one of my best gifts to OPR since I've left. She's amazing. Wow. Well, thanks for having me, and thanks for coming this week of Thanksgiving. I didn't know that turnout would be so great with people headed out of town, so I appreciate you coming. And it's really great to be back here at OPR. It's a bit like going back in time for me, so it's a real pleasure. And my work since being here as a graduate student has gradually drifted a bit towards more biological questions from social science questions. Still a mix of both, but the questions I ask are still really grounded in the training and perspective I received here at OPR. And I would just like to say that that training has continued to be really unique and a strength in my work in different disciplines and in different environments. So it's something I hope all of you grad students are really relishing, and I encourage you to enjoy your time here. It's really a special time. So today I'm going to talk to you guys a bit about a series of papers on a project that's been funded by an exploratory R21 from NIH and also a recently begun R01 grant. And this is largely in collaboration with Allison Aiello, an epidemiologist at the University of Michigan. And this, as you can see, is an ongoing research agenda. We've started to explore some interesting associations, but I would definitely welcome any feedback that you guys might have and suggest promising directions for future research from your perspectives. That would be great. So I'm going to talk about a few of my overall research aims before I dive in to the research. I had to sit back and think about sort of what my larger scale aims were in tying this all together. I think one of the first ones is to better characterize human immunity at the population level, and I'll talk more about each of these a little bit in more detail. Then from the perspective of thinking about population health and overall trends in morbidity and mortality and aging, I'd like to explore the role of immune function in population health. Is it important? Is it not important? How much should we try to understand this particular physiological system? And then third, the work that's more grounded in the socioeconomic differentials work that I did as a grad student is to think about how the social environment, including of particular interest to me is social stress, shape immune function over the life course. And then ultimately the goal is to bring that back to the literature on social inequalities in health and think about how much immune function might help us explain the social differentials in health that we see across populations. Yes, and this I just wanted to show you guys to say that we're not alone. This cartoon is about the microbiome, which the Human Microbiome Project has pretty recently started publishing papers. I'll talk a little bit more about it later, but just to remind us all that there are 10 times more microscopic bacterial cells in our body than human cells. So they outnumber us by quite a lot. And I just wanted to point this out to kind of introduce the idea that even though we think in the epidemiological transition that we've passed through the age of infectious disease, microbes are still a very important part of human health. And this is what has led me to want to study microbes in the immune system a bit more, is that it's something that we really continue to co-evolve with, that has been part of our physiology for millions of years, and we're kind of our own ecosystem of good and bad microbes, and so it's an important part of human reality. But that leads me into why am I so interested in immune function. I know from the social science perspective that might seem pretty narrow, and it definitely is. But I'd like to talk to you a bit about why I have found it intriguing, why it drew me to study immune function more, and hopefully kind of intrigue you guys. Maybe you won't find it as fascinating as I do, but I think there's a lot of interesting research going on that can potentially bring this back to social science as well. And the main thing that has fascinated me is learning about all the different types of immune processes, including inflammation, which you have perhaps heard more about in the news, but that interact with lots of other physiological systems in important ways. If you think about metabolism, for example, some of the work to come out of the recent work on microbiota, so the gut bacteria that we all have in our stomachs, that you acquire pretty early in life, possibly through the birth canal and breastfeeding and early diet, but there's other sources of variation in the microbiota. These things have been tied to obesity already. There was different microbiota found in lean or obese humans. Now, of course, that could be due to a lot of things, the stuff people are eating. So they took sterile mice and experimented with giving them one version of the microbiota, and they became obese, and the other ones stayed lean, et cetera. So there does seem to be something about gut microbiota that changes how the calories are harvested from the gut and how those are changed into energy and stored in the body. So we see these associations between gut microbes and metabolism. Then I thought this was an interesting example of the other direction. Obesity has been associated with impaired immune response to influenza vaccine in humans. And with the H1N1 flu epidemic that we had a couple years ago, I don't know if anyone noticed, but for the first time, obesity was one of the risk factors for mortality with the flu. There was a lot more deaths in patients who were obese. So there's sort of this bidirectional association between immune function and obesity that's potentially getting more important as the obesity epidemic continues and people live a longer number of years obese as well. There's obvious links kind of with the hygiene hypothesis between the immune system and allergies and asthma. One that is of particular interest to me are links between infections and mental illness and cognition. Some of you may have heard about toxoplasmosis, the kitty litter disease. It's a bacterium that, again, a lot of us are exposed to, and there's no problem with it, but there's been ongoing literature about toxo and schizophrenia. But more recently, there's been interest in looking at sort of more a wider variation of behavioral changes that might be associated, because in rodents, so rodents who carry toxoplasmosis, the bacteria is actually very clever in trying to maintain itself and get reproduced. And so it changes the rodent brain to, in the fear and anxiety regions, to not be afraid of cats and actually be attracted to cat urine. So this obviously helps it be successful in getting eaten by cats and reproducing in the cat gut. But there's been some evidence. So we ingest this, and it wasn't designed to do that in us, but we share obviously some neurological pathways in an evolutionary sense with other mammals. So people have been looking into how this affects anxiety and fear and risk-taking behaviors in humans. So that's a connection I find interesting. And then just a recent article in the New York Times really highlighted the potential links between autism and as an immune disorder, and particularly looking at inflammation exposures in the womb. And then just to throw out the links between infections and cancer, you probably know about HPV and cervical cancer, hepatitis, viruses, and liver cancer. H. pylori, which causes ulcers, has also been associated with gastric cancer. So there are links to other chronic diseases as well. But in general, there's a lot of bidirectional communication between the immune system and other physiological systems, and especially the brain, which I think is kind of the leading edge of the stuff that we're starting to learn about, that I find really interesting and hope that will intrigue you guys as well. So my first goal is really an overarching one that I hope through the course of lots of research to start to accomplish, but we really don't know much about human immunity in a descriptive way at the population level. And this is really a limitation in part of the clinical measures that we have. If you think about going to the doctor and you want to get your cardiovascular risk factors, and they take your cholesterol and your triglycerides and your blood pressure and maybe your waist to hip ratio, they can come up with a pretty good summary measure of your cardiovascular or metabolic risks that have been validated to predict heart disease and things of that sort. But there's really no such markers or parameters that exist for immune function. And this seems to be a limitation on just the clinical side, that we haven't figured out what those good summary indicators are yet. And there's a lot of work being done, I think, to figure that out, whether it's trying to come up with a set of 5 or 6 biomarkers, possibly inflammatory cytokines. There has been some markers coming out on inflammation or proportions of T cells, something that might help us characterize immune function. But since we usually get those from the clinical side, this is also something that hasn't really been explored then in biodemography and population health. We've started to explore variations in inflammation across different populations and different settings, but we don't really have a lot of good markers the way we do for traditional risk factors in cardiovascular disease and metabolic health. So as time goes on, and hopefully more of these markers are included in population surveys, we'll simply get a better sense of what immunity looks like across different populations and across time. So my second research goal, and it's sort of one strand of my research, is to understand the role of immune function overall in population health and aging. And one parameter that we do have to explore as kind of a surrogate marker of immune function is immune response to persistent infections, in this case, latent herpes virus infections. And so a lot of you, if you don't know all of these viruses, you probably have some association with herpes as not a good virus to have. And that would be, that's true, but it's not really these viruses. It's in the class of viruses that I'm talking about. That would be herpes virus simplex 2 is genital herpes, the one you guys are probably thinking about. But all of these herpes viruses are in the same class. And these are very common, very prevalent in the population. So it's likely that a lot of you have most of the rest of these viruses, and it doesn't say anything bad about your character if you have them. So cytomegalovirus, CMV, is one that I work with a lot. And this, again, is mostly acquired early in life. You probably would never know you had it. You might have a mild fever, might be totally asymptomatic. It's only really dangerous if you get it as a primary infection during pregnancy. It can cause congenital defects, and it's also very dangerous if you have HIV or compromised immune system from organ transplants or chemotherapy. But normally, a lot of us have this and wouldn't know it. The same with Epstein-Barr virus, which causes mono, you might be familiar with. And again, it's not, most of us probably have it and didn't get mono. It really only causes mono if you don't acquire the virus until adolescence or early adulthood. It's much more severe primary infection. HSV1, herpes simplex virus 1, causes cold sores. You might be familiar with that one. And then varicella zoster virus is otherwise known as chickenpox, and then shingles later in life. So the interesting feature about these viruses compared to the common cold or other infections you might get is that your body never completely clears them. They stay with you really for your whole life, and your immune system and your body have to continuously work to keep them under control, and that's why they're so dangerous in the case of immunosuppression. But with that, the benefit for measurement of immune function is that we can use antibody response to these viruses as an indication, an indirect measure of immune function. So if your immune system is doing a good job keeping these under control, then actually antibodies to these viruses are low. It's if your immune function starts to decline because of stress or any of these other conditions like chemotherapy, then the virus starts to replicate again in your system, and then your immune system has to come and produce antibodies again to defeat the virus. So this latent reactivation of the herpes viruses and the subsequent immune response has been used across several different disciplines as an indirect marker of immune function. And the most prominent literature in which these viruses have been featured is psychoneuroimmunology and a lot of psychology literature that has looked at, in sort of small samples, associations between stress and immune function. And I don't know if any of you know this psychology literature on stress and immune function. Ron Glaser and Kikolt Glaser, his wife, have done a lot of interesting things where they put husbands and wives together, and I'm not sure how they induce them to fight, but they give them a wound first, and then they induce them to have a good interaction or a bad interaction, and they've measured rates of wound healing and associated that with social relationships, and they do a lot of interesting experiments. But they've done a lot with these herpes viruses and found that they were strongly associated. So increases in these antibody levels have been strongly associated with bereavement. They've looked at medical school exam takers, cadet basic training at West Point, kind of isolated stressors, also some more cross-sectional type of chronic stressors, caregivers of spouses with Alzheimer's, poor marital quality, other reports of chronic stress. CMV in particular that I've been most interested in has been looked at with academic stress. Also, NASA has looked at it with space flight, because you can imagine going into space is kind of an extreme physical and psychological stress. And there's been strong relationships found with these herpes viruses. And I thought for family demographers, you might appreciate the tie-in here with union formation and dissolution. It says, "You've broken down my immune system." I thought that was fun. And this is just an example of looking at that change in antibody levels over time. In this case, these were the exam stress studies. The beginning of the semester is the first point, then there's 2 exams in different subjects, and then after the end of the semester, you can see the antibodies kind of go back to even lower than the beginning. So that's sort of what these studies typically look like. They're very kind of specialized samples and small, discrete stressors. Biological anthropology has also utilized this as a stress marker, particularly the EBV, Epstein-Barr virus titers, and work by Tom McDade and a lot of his colleagues. They found associations with higher EBV and increased life strain in adolescents from the Great Smoky Mountains, higher lifestyle incongruity among indigenous Siberian herders, which I had to read the paper to understand it's some kind of marker of extreme cultural and economic change, sort of rapid cultural change. Family stress in Kabul, and perceived discrimination in Latino immigrants. So this has been used in a variety of contexts in anthropology as well. So these latent herpes viruses seem to be picking up associations with stress in lots of different contexts, and that was immediately appealing to me to think about using it to examine interactions of the social environment and stress and health. But beyond just being a marker of stress, I found these infections really interesting because of some direct relationships with health as well. And so that's what I'm going to talk about first, is building that link a little bit more. There'd been some suggestive evidence of lots of different infections and chronic disease, but CMV antibodies in particular have been associated with cognitive decline longitudinally in the elderly, so higher antibody levels at baseline, faster cognitive decline. I'm involved in a recent fMRI study doing imaging of the hippocampus and finding lower hippocampal volume with HSV1 and CMV infection. And then CMV has also been looked at a little more broadly in epidemiology to look at mortality, frailty, and immunosenescence, which I'll talk about, sort of age-related immune decline. And then lots of different of these infections have been implicated in cancers, as I mentioned, some associations with cardiovascular disease, sort of inflammation of the arteries. CMV has actually been found, viral DNA of CMV in Alzheimer's, plaques, and dementia. So there's some interesting suggestive associations there. But the one that's really kind of compelling in thinking about aging and mortality and whether this might help us understand patterns of morbidity and aging across populations and different social groups is the association of CMV with immune aging, which is also known as immunosenescence. And there's been a lot of work in immunology that's looking at this. Again, a lot of it's just in vitro or in very small human samples. But the assertion is that CMV is more the driving force behind age-associated alterations to the immune system. So you know when you get to older ages, you're not as good at fighting infections. You're more likely to die from things like pneumonia that wouldn't take you out when you were younger. There's a sort of overall frailty of the immune system. And it seems to be that actually CMV infection might be more strongly associated with that immune aging than age itself. And this has been looked at specifically with proportions of T cells. So CMV causes this increased proliferation of T cells in the immune system that are specific for CMV. So it basically keeps replicating and accumulates T cells specific for that infection alone, taking up kind of the immunological space and making it harder to fight off novel pathogens is essentially what's going on. And it seems to be the one infection that humans have that does this the most. So in fact, by the time you get to be at older ages, a large fraction of the immune system is dedicated almost entirely to CMV. So if you think about that as a single pathogen, 50% of all CD8 T cells, it's a really high proportion for a single pathogen, and that's why it's getting a lot of attention. The question is, does this have clinical relevance? Is this important for human health? That's just starting to be explored a bit. CMV has been associated with reduced immune response to influenza vaccination among the elderly, and that's a very important indicator that they're not mounting as good of an immune response. But interestingly, this was also recently found in a younger population of college-age students getting vaccinations, and I found that really interesting because you might think this immune decline would only start to be important in old age, but the fact that we're seeing these big changes in younger populations is really interesting.
Is it because of the nature of the virus?
The virus.
That happened with the Biden one, I think, that there was more, the younger people were more vulnerable. Wasn't that the case with that one?
Yeah, that's true. They were, there was some indication that they were mounting more of an immune response, which was more dangerous. So, yeah, no, that's a possibility. No, that's a good point. So it's a bit of a puzzle. So CMV seems to be an intriguing part of two processes that look very similar. One is the immune consequences of chronic stress, which is of interest to some of us in social epidemiology and social stress, but also the immune changes associated with aging. So the question is, could CMV possibly be a common source of these two things? And this is just a list of some immune changes that happen with psychological stress. The T cell proliferation is really just increasing an immune response to a novel pathogen. It's decreasing your ability to do that, lowering your response to vaccination, increasing viral activation of those latent herpes viruses that I talked about, increasing inflammation, which is bad for lots of things, and decreasing telomere length, which I'll talk about a little more later, which is the tips of chromosomes, which you guys might have heard about in biodemography or in the news. And as they divide, cells divide, telomeres get shorter, and when they can't divide anymore, the cell will die. So telomere length has been used as a marker of cellular aging. And so immune system aging looks a lot like these same phenomenon that happen with psychological stress. So it might be that they share a mechanism. And then, as I mentioned, these memory T cells that accumulate that are specific for CMV, the big feature is that you're accumulating these late differentiated T cells that can't respond to any new pathogens. They're sort of already committed to fighting this pathogen, and so they're not that great for the immune system to accumulate these late differentiated cells, is basically the idea. But we see a lot of similar features that with CMV driving a higher proportion of those cells, we see a lot of similar features with the immune aging and the chronic stress features of immune function, including shorter telomere length. So just that far right column is just kind of the most differentiated T cell that's kind of most committed and not able to go back and fight other pathogens. They have shorter telomeres, these tips of the chromosomes. And so the question we're trying to probe is this infection a common determinant of what we see as the immune effects of stress and aging? And the conceptual model sort of looks like this. We know a lot about psychosocial stress impacting immune risks and T cell senescence. But what we think might be going on is that CMV reactivation is an important mediator of that association, and that ultimately this is what's leading to all these biological outcomes that we care about as far as aging and morbidity. And then later on, I'll put sort of the social status part up there as something I've been interested in, looking at the association between social status, stress, and these immune pathways. So while most of the work on CMV and immune health has been done in these small samples in immunology or even in vitro, we wanted to take a stab at looking at whether CMV was associated with health at the population level. And there's, it's not in that many surveys, so we've tried to take advantage where we could. But we did have data from NHANES 3, which the survey ran from 1988 to 1994, and we had follow-up until 2006 for death ascertainment. And these were adults 25 and older. And for those of you not familiar with NHANES, it's nationally representative of the US. And we found a significant association here just between CMV seropositivity and mortality over follow-up. I think the hazard ratio, when it was all adjusted, was about 1.19, so it's not huge, but an interesting, adjusting for as many other comorbidities and social factors as we could, we still found this relationship. They're 25 and all the way then up to 90s in NHANES.
What happens if you look
Only at the older ones? You start to lose a little power because so many people are positive by then, but I think you still see the association. But I'll show you sort of how seropositivity goes up with age in a minute. Just whether you have the virus or not was interesting, but because seropositivity gets so high, it's a very prevalent virus. What we were really interested in is this reactivation. Once you have CMV, the sort of losing control of it with your immune system, not being able to keep it at bay, that's what we think has all these poor implications for immune function and immune aging. So that was not available in NHANES, but we were able to look at that in an epidemiological study called SALSA. It's the Sacramento Area Latino Study of Aging. It's the best study acronym ever. Best acronym. So you can figure out sort of the study sample from the name, elderly Latinos in Sacramento. But in this case, we didn't see a sort of a linear relationship between CMV antibodies in the sense that being in the lower three quartiles, the risk wasn't distinguishable from across those, but having the highest level of antibody response, the top quartile, was associated with higher risk of mortality. So we found this somewhat supportive of it contributing to this general immune frailty. So those were interesting, but we wanted to kind of look more directly at how CMV might be affecting mortality and aging. And one pathway that we were able to look at is these telomeres that I mentioned earlier, these tips of chromosomes that have been used as a marker of cellular aging. And so again, your cells divide and telomeres get shorter and shorter. They can be repaired and lengthened by an enzyme called telomerase. But once they get to a certain length, they sort of lose genomic stability and the cell dies. And then obviously across different organs or systems, that can lead to poor health outcomes. There's been sort of a boom of research on telomeres that you might have heard about in the news, and I'm sure it makes a lot of the health news. It's been linked to, so shorter telomeres have been linked to mortality and things, and this is all being measured in the blood, so leukocyte telomeres. But really, your telomeres are in every single cell. So the telomeres in your blood may or may not be telling you about the telomeres in your brain or heart tissue. But those are things that we can only get, obviously, with difficulty or from autopsy. So in practicality, most people use blood. And Alyssa Eppel, you might have heard of a lot of her work linking social stress, stress of caregiving, and other forms of life stress to shorter telomeres. I think the literature on socioeconomic status by itself and telomere length is a little mixed, but there have been some findings of an association there. But a lot of this literature looking at stress and mortality has started to include telomeres. And we were really interested in thinking about how CMV might be involved in this, not only because there's a relationship between CMV and stress and CMV and mortality, but because of the very particular thing that happens with CMV in the T cells, where it drives the proliferation of all these certain type of T cells in the blood, which make up a lot of this leukocyte telomere length. These highly differentiated T cells that I talked about that kind of indicate an older immune system all have shorter telomeres, just mechanistically. The naive ones have longer telomeres. These short ones that have been influenced by CMV end up having shorter telomeres, sort of in vitro. So we thought, given the associations between stress and CMV and stress and the shorter telomere lengths in T cells, this might be important in explaining broader variation in telomere length in individuals. So we know telomere length decreases with age, but there's still a lot of variation that we don't understand. For a given age, there's a lot of variation in telomere length that we can't explain. So we wanted to understand whether CMV infection was really playing an important role in these studies looking at stress and telomere length and telomerase activity. So we had data from the Whitehall 2 study, which from the UK, some of you might be familiar with. And this was a subsample, the heart scan study, where they did a lot more intensive biomarkers and stress reactivity tests, and then also happened to measure telomeres. And we added a panel of infections for which we only have CMV data right now, but intend to look at a broader array of infections. But so this is just recent, pretty basic preliminary stuff that we've done looking at whether CMV was associated with telomere length and telomerase activity. Obviously, we thought that it was, and we wrote a grant hypothesizing that it was. This was our result for CMV and telomere length. So this is actually continuous antibodies. So seropositivity was not really related to telomere length. It was in the expected direction, but not significant. But we wanted to sort of look at this immune activation part of it. So you can see nothing really going on. Maybe the highest antibody response is a little bit lower, but not significant. So that just goes to show you can't always support your hypothesis even from a big grant. But it's good to know that at the beginning of the grant. No, but actually, this was an interesting finding then. So telomerase, as I said, is this enzyme that can repair telomeres, make them longer, but it also, beyond doing that, also protects them from this genomic instability. It can help them continue to be viable and replicate longer by increasing their stability. So we did find an association between higher CMV antibody response and lower telomerase. And that was robust to controls for employment grade and which wasn't actually associated with telomeres and telomerase. But so we thought this could be some indication that CMV does play a role in some of these telomerase dynamics, but it doesn't explain really why CMV was not associated with telomere length cross-sectionally. Some people think that telomere length change over time might be more important to look at. We can't look at that yet, so cross-sectionally we might not be seeing it. And age was actually not associated with telomere length in this sample, which is very unusual. In almost all other studies, age and telomere length are strongly associated. So, so we don't quite know what's going on there. It might be something special about this sample. But we did find this interesting finding that telomerase activity seemed to be downregulated for those with high CMV antibody levels. And so this could be part of the process that CMV is contributing to senescence of cells and contributing to aging and frailty more generally. But we're going to be adding more infections, and this is really kind of just the beginning of this project. So the benefit is we have the whole Whitehall data with sort of a lifetime of other exposures, lifetime stressors, lifetime socioeconomic history to look at with this, so it should be an interesting project. And getting to the part that you guys might be more interested in, my third research aim has been to think about then, since we have found at least pretty suggestive evidence that these infections and immune response are associated with various health indicators, how does the social environment shape immune function over the life course? And this is very much motivated by questions that I studied here at OPR, and thinking about this social gradient in health, which I'm sure you guys know well, that wealthier, more educated people live longer, healthier lives, and sort of our traditional risk factors don't seem to explain that. We could have a lot of discussions about that, I'm sure. And then I was very drawn to looking at biological pathways linking socioeconomic status and health, and there's been a lot more of that going on, a lot more surveys including biomarker data, but as I said, we still don't really have a good sense of how immune function fits in. Again, inflammation is one area that has been better covered, but overall, we don't know a lot about immune function. But what again attracted me about thinking about this system was the association with stress. You guys have probably heard a lot about stress as a potential pathway linking the social environment to health outcomes. The hypothesis being that lower SES is associated with greater exposure to stress. You might not think that when you're a grad student at OPR or in your first jobs, but in general, I think we'd probably agree that exposure to things like financial stress and job insecurity and low job control, unsafe residential environments, discriminatory experiences, daily hassles, or if we think of even just vulnerability to crises such as the storm that we just had, having fewer resources to fall back on would make that a lot more stressful. And the hypothesis then is that it's the physiological effects of this stress that kind of lead to the earlier aging of the poor, was a title of one of Eileen Crimmon's papers that really captures this idea that we're accelerating the aging process through these extreme chronic stresses. And Bruce McEwen and a lot of other researchers have focused on cortisol, the stress hormone, as the potential explanation for this. And that was something that I was interested in for a while, and did a review that's now a few years old, 2009, looking at, so what's the evidence for associations between socioeconomic status and cortisol? And it actually turned out to be really mixed. Some found higher levels, some found lower levels, but it wasn't very systematic. I'd say since then, the measurement has gotten better. We've learned that we really need to follow people over the day and get a sense of this diurnal pattern. And I think that is starting to be more consistent, that lower social status or disadvantaged groups tend to have this blunted response, cortisol response. and instead of an overactive cortisol response. And that, has been hypothesized kind of as burnout. If you keep turning on the system a lot early in life, you might then not be able to respond when you need it. But anyway, the cortisol, is hard to measure because you do have to capture this diurnal rhythm. And that was one thing that led me to kind of seek out this other indicator, this, leveraging this link between stress and immune function. So I talked about these links and thinking about it as a biomarker. In contrast to cortisol that has a diurnal pattern, you don't have to worry about that. If you measure it, it's going to be stable over the course of a day, certainly. And it's highly prevalent and easy to assay in blood or saliva. So this was our general framework in thinking about how socioeconomic status and social factors might be related to immune response. We're really interested in how social factors influence exposure, because obviously, if living with these infections a lot of your life is more dangerous, since you never get rid of them, your immune system, if it has to struggle with them for a greater number of years, that might be detrimental. So we're interested in when people get these infections. It's likely that social status influences that. Crowding, we can think about it in developing countries, it makes more sense, but even in developed countries, there's probably a lot of social factors that influence exposure. Then given exposure, how do social factors influence susceptibility? We can all be exposed to the same virus and we won't all contract it, so how do things like stress, nutrition, other health behaviors affect susceptibility? And then obviously we're interested then ultimately in these persistent infections and how immune response plays out over the lifetime with the interaction of stressors, other comorbid conditions, re-exposure, et cetera. And thinking about how socioeconomic status might affect susceptibility, it's really hard to get at in typical survey data, but Sheldon Cohen has done some neat experiments, which I'm very jealous of being able to do this. I don't know how he talks people into exposing them to the cold virus by shooting it up their nose, but he does. I'm not sure what the payment is that he gives people, and they're all college students, I'm sure. But perceived social status was significantly associated with, so given the same exposure, everybody gets it shot up their nose, literally. Whether you actually contract the cold was associated with socioeconomic status and sleep and lots of other things, but it does indicate there's some scope for susceptibility by social factors. And we were interested in this, looking over the life course, partially based on the hypothesis by Crimmins and Finch, looking at sort of the lifetime infectious and inflammatory burden as a pathway that shapes later life patterns of aging and mortality. So the more infections possibly you have earlier in life and the longer you deal with them might contribute to this inflammation burden. So we wanted to take that and see, what does the distribution of these infections look like by socioeconomic status, first of all. These are really common infections. You get them kind of like the common cold. You wouldn't necessarily think they'd be distributed in a very systematic way, the way you might, tropical infections. But here's what we found for CMV, seropositivity, and NHANES by income. So again, this is 1988 to 1994. But you can see how the seroprevalence really increases with age to where almost everyone has it at 75 and above. But there were big differences in seropositivity at different ages by income quartile, and we found something similar for education. And so this is just interesting, especially if you think that for these latent viruses that possibly the amount of time that you have them affects your immune function over time. Because obviously this is cross-sectional, so you all know it's not following a cohort, but if we thought that people with lower income were likely to acquire these earlier, they're going to live a greater proportion of their lives struggling with their immune system, struggling with this infection. We looked at this for a series of other infections in NHANES. We tried to, so NHANES really is kind of like a surveillance, like measuring a lot of infections just to see what's the prevalence in the United States, and not necessarily for our research purposes, but we kind of picked the infections they had measured that were linked to chronic disease in some way. So H. pylori, again, is the peptic ulcer and gastric cancer. Bacterium, hepatitis. B is HPV. And these other two down here are periodontal pathogens. So stuff, you might have heard of the association between, bacteria in your mouth and heart disease. So that sort of idea. But again, just kind of descriptively looking at this, we see pretty consistent associations with having the infection and across race, ethnicity, with blacks and Mexicans, Americans having more, but also within those groups by education category. Not all of them, but we see pretty consistent patterns for the more prevalent infections. We also looked at this in kids in NHANES, and in this case we took those infections and kind of made a latent burden of infection with a factor model, kind of trying to capture this idea that we're not measuring the true burden of infection, but these are imperfect indicators. So these are factor scores. And again, just a similar pattern, just to say that minorities in the U.S., kids aged 6 to 16, with the sample, are more likely to have more of these infections. And also by parental education, there are differences. This is sort of hot off the press, Epstein-Barr virus. This one, herpes virus, was just released by NHANES. They did some surplus samples. And so this is EBV. So we didn't really know what this would look like. We assumed it might look similar to the other infections. But these are kids aged 6 to 19 in NHANES, more recent NHANES. Just logistic regressions for seropositivity. About 66% of these kids were seropositive for EBV. And you can see just the strong associations by race, ethnicity, and the likelihood of having the infection, by whether you're below the poverty threshold, and parental education. So that's very similar to what we saw with the other infections. But again, this is one of those latent, lifelong infections that stays with you, has been linked to some cancers and chronic disease. So just from this descriptive data, it was very clear that low socioeconomic status and race ethnicity in the U.S. are associated with more likely and earlier acquisition of several common infections. And so it seems like the overall burden of infection in both kids and adults in the U.S. does appear to be higher. And so it's possible that, this isn't just a phenomenon of past cohorts where infections were really important, but it could be that differences in early infectious environments are still relevant for longer-term health outcomes in explaining social differentials. So those were just whether you had these infections at all. But as I talked about before, we were really interested in this immune response for those who already have these infections as a marker of decreased immunity and possibly getting at this social stress aspect that we weren't as able to get at with cortisol. So we were able to look at continuous antibodies of CMV also in NHANES 3. And here we found, I was kind of surprised it was even this evident, but we found strong associations. There's a similar figure for income across, so you see how antibody levels increase with age, and this is what the immunologists find and would predict. So you sort of lose immune control as you age. But you can see that the level of this really varies by education. And if you just do the quick and dirty kind of look across the figure, you can see that sort of the highest level of education has, the immune response of someone 20 years younger, and in the lowest category of education. And we often hear numbers like that thrown out about this accelerated aging, across social class, but sometimes you will hear something like 10 or 20 years of accelerated aging. So if this is a surrogate marker of immune function, we did see something like that. Obviously, this isn't in a lot of, these markers aren't in a lot of surveys yet. This was cross-sectional. So we tried to control for other chronic health conditions, health behaviors, to try to really isolate whether this might be related to social stress. But we can't rule out that this CMV was related to some other latent burden of disease that we weren't measuring that's suppressing immune function. The stress pathway is still speculative, but that was one reason we then decided to go on and look at kids. The theory being that looking at this life course perspective, that early experiences are thought to model immune function, but they're probably going to have fewer of these confounding chronic diseases to confuse the situation. So there'd been a little bit of work looking at early childhood maltreatment and HSV1 titers, but we looked at this with continuous antibodies in NHANES and found that just being below the poverty line, there was not a linear association with income, but being below the poverty line was associated with higher antibody titers, when most other things were not. We tried to account for other things that would suppress immune function, being low birth weight, recently ill, or having asthma, but we found this association with the poverty line. And so EBV has recently been added to AdHealth. Wave 4 of AdHealth, if any of you are using that data, has just released EBV. So we've done some preliminary looks, and also at the continuous antibody titers in NHANES. And so interestingly, this is sort of the same type of analysis with kids in the same age range. We did not see an association with continuous antibodies. Remember, we saw big associations with just having the infection or not. But for this continuous antibodies, we don't see associations with EBV and poverty, in contrast to CMV. So that's kind of a puzzle that we'll have to think about. In AdHealth, we're looking at stressful life events and how they predict EBV. And we do find associations. And these are things sort of tallying things like the death of a parent, death of a sibling, injury resulting from attempted suicide attempt, having witnessed violence, victim of violence, those sorts of stressful life events. As far as the SES measures here, we really only see this category of college education being significantly associated with EBV compared to high school. So I'm not sure how big the graduate degree category is, if that's surprising, but we see stronger associations with race, ethnicity, and higher levels of EBV titers indicating this impaired immune function. We were really interested to see if stressful life events were mediating any of these relationships between SES and EBV. You could kind of argue that this declines for women, but not so much over here for men, and not so much in the race ethnicity categories. But this is, again, very, very preliminary work. But for any of you who use AdHealth, I would encourage you to look at the new biomarker release. They have EBV as well as some markers of inflammation, if this has piqued your interest in any of those associations. Okay, sure. Okay, I'll kind of skip over this. This was just a study that showed that social status actually interacted with the effect of CMV on the immune system, which I can tell anybody about later. So, yeah, you can just read this and we can talk about it later. But so sorry I went on too long, but thanks, guys. Great, yeah, I'd be happy to take questions about anything or where you see this going, or if this sparked any of your interest.